Today I'll be talking about how we, as geneticists, try to deal with improving heat tolerance in pigs. And I'll bring to the table a new tool, gut microbiome, that we've also been working on for a while on different traits, but perhaps we're close to come out with a potential use for this tool in improving heat tolerance as well. So today I'll report a little bit of that. I'll be speaking also on behalf of Dr. Christian Malteca, who have been working closely on this topic for the past 5 or 6 years. Let's start with an outline. First of all, I'll review a little bit of the work we've done on heat tolerance and selection for heat tolerance using genomics. And then I'll review the work we've done on the use of gut microbiome in pork production. And then I'll try to link the two things with a new study I'll present on connecting gut microbiome and heat tolerance and genomics, of course. We don't leave it behind. I should say that both myself and Christian Malteca are geneticists, so we have a different view of the problem, but I'll try to take any questions and actually also to present the topic from a different angle that is not just about breeding and genetics. First of all, genomics of heat stress. We just came out within the last 12 months with 2 papers. The first in their kind, because there were not so many studies like this out there. We were able to work on the potential of implementing selection towards improving tolerance in pigs. And there was both in sows and in growing finishing pigs at the commercial level. The first study involved 4 populations from both SPG and the mash shows for a total of 120,000 liters. They're all nucleus herds of maternal lines. So 4 populations, there were 2 lines over 2 companies. We also had access to a large amount of genomic data with about 15,000 genotyped individuals. Again, these were nucleus individuals. The graphic at the bottom is basically the main take-home message, which is a plot of how total number born will change, actually will decrease as the humidity increases. In this case, the humidity was collected using the weather stations out there. But I'll show you later, we're also moving with collection of climate data inside of the bar. But even with this zero-cost study, we were able to identify individuals that were less tolerant. It will be the red lines, so show a stronger decline in total number born as the humidity was increasing, but also individuals in the blue line that were fairly tolerant. They wouldn't really change their performance. What I'm showing here in this plot is basically breeding values. It's very uncommon to show breeding values as lines, but this is what we can produce. We can basically produce a breeding value for each individual, for each single value of humidity or temperature or THI, depending which one has the most impact. Like this, we can select for the tolerance itself by identifying the blue lines or the red lines, or also selecting for for adaptation to a specific climate. Oops, I went back instead of forward. Because of the large amount of genomic information, we're also able to identify the genomic regions that were underlying these traits. And in general, we are able to produce very accurate breeding values. So we could say that we'll be ready to implement genomic selection for improved heat tolerance in sows for traits like I showed, total number born alive, and birth weight. We also move forward studying growing and finishing pigs in a commercial farm. This data was courtesy of the mash shows. We had about 130,000 commercial pigs out of their commercial tester herd. We had their sires, the purebred Duroc sires genotype, about 500 of them. And basically the deliverable was the same. We were able to identify the individuals that even in the grower finisher are more heat tolerant or less heat tolerant for, in this case, carcass average daily gain, but also for back fat and low in depth. And once again, we're able to select potentially for individuals that will not see a decrease in growth during the summer months, allowing to reduce the seasonality that affects the systems. as a take-home message of this part of just repeat what I've said, that we're ready to implement genomic selection for heat tolerance, both in maternal lines and terminal lines, as we select them for the cross-breed performance. Let's move into gut microbiome. We've been working on this topic for a few years now. We were invited to submit a review about 2 years ago on this topic and how it can be used in breeding programs for improvement of efficiency, especially feed efficiency. That's what we're working on most for now. And overall, let's see. Okay. Overall, we'll try to answer some very simple questions. When we use gut microbiome, so the whole ensemble of species, mostly bacteria that inhabit the gut, what traits are affected by this composition, right? Different pigs will have different composition of the ecosystem that inhabit the gut, and how does that reflect on the traits of interest? Of course, it reflects on gut health, as we've seen in our presentation this morning, but ultimately on the commercially relevant traits, which are the ones that are mostly affected. Also, once we see the overall impact, are there some species that have more impact than others? And if yes, should we actually focus on these species? And ultimately, as we are geneticists, can we use gut microbiome breeding programs? We leverage on 2 trials, Argo quick. We had about 1,200 commercial pigs, again, courtesy of the Mashoffs, that were followed through the whole process, were phenotyped for meat quality, growth, back vestigial traits. They were genotyped, and also gut microbiome was sequenced at 3 time points, weaning, on test, and off test. Another experiment was done in collaboration with Smith & Freeman Genetics in their nucleus farm, where the 3 breeds are put on the 5 feeders. So we have feed efficiency, feeding behavior, and microbiome together with the genotypes. And what came out in a nutshell is that, well, first of all, we can find a number of traits that are affected by microbiome more than others. And there's the new term that came out is microbiability, just like we had irritability. Now we have microbiability. It's the same concept. Instead of having the genetics of the animal affecting the phenotype, we have the genetics of the bacteria that inhabit the intestines as affecting the phenotype. As I mentioned, some traits are more affected than others. On top of the list, there is fat deposition. That's the one that shows the largest impact from gut microbiome. Then, surprisingly, we had meat tenderness, for example, slice shear force, or also subjective measures, followed by feed efficiency, and feeding behavior, as it wouldn't be so surprising. And then at the end, also carcass growth and muscle deposition. To give you an example what it looks like when we try to include gut microbiome in our models, well, just consider this is a simple irritability estimate for fat depth, right? The blue bar is the irritability. In this case, it was fairly large, 40%. And then we include in the model the microbiome as a whole as measured at weaning. That would be the green thin line. Not a lot of impact as the weaning microbiome, right? That could be expected. As we move to the on-test microbiome, so it gets closer to the time of actual fat deposition in the pig, the impacts become large. And when we move at the off-test microbiome, so basically at the end of the 6 months trial, the impact of gut microbiome is sizable. It's almost as large as the irritability. Actually takes away some of the irritability. So this means that if we want to understand that trait overall, gut microbiome can provide a lot of information beyond the simple breeding use. And I'll go back on this. We also identified some species that were associated with some important traits. And here, there's a list of them here. I want to highlight lactobacillus that was strongly related to feed efficiency. The surprising thing is that lactobacillus depends on the diet, right? The amount of fiber in the diet. And we found that it was differently abundant between nucleus and commercial farms. But in both cases, there was a strong association with feed efficiency, which means that doesn't really matter how much we change the lactobacillus abundance, it will still show some variability and will still show some impact on feed efficiency. And this, I believe, could be important as we move forward to use gut microbiome as a biomarker, for example. We want to have a measure of feed efficiency. Well, we may just measure the relative abundance of lactobacillus, and we could have a good estimate of the feed efficiency and digestibility in the pig. One last thing, as a breeder, I had to put it out there, gut microbiome is heritable, or is partially heritable. So despite we can change the microbiome, despite the fact we can change the microbiome with the diet, and the microbiome also changes from one pig to another because of reasons that we don't truly understand, gut microbiome is heritable. So we can select for an individual that has more or less one species, depending on if it's desirable or not. But also, as we have been selecting pigs for their ability to grow lean muscle, be more feed efficient, and so on, we have also been selecting for their gut microbiome. So the gut microbiome nowadays pigs is definitely different than the microbiome that was there in pigs 20 years ago. And this is just a plot to show the heritability, like I said, as a breeder, I had to put it there. And one interesting thing is that as the animal matures, the gut microbiome becomes more heritable, which means that the animal can control the gut microbiome because of its genetics. But that doesn't really happen at weaning. In fact, at weaning, the animal is more exposed to changes and is less resilient. As the animal grows, becomes more resilient, part of this resilience could be due to the establishment of a solid ecosystem in the intestines. Because of this connection with resilience, and because of this connection with gut microbiome modulating the pig and its physiology, well, at some point we decided, well, let's connect the two dots and let's see how gut microbiome can help improve heat tolerance. We actually got invited to submit another lit review. It's just be submitted on how we could use gut microbiome as a biomarker. Our student Yushin He has just prepared this literature review. And there is the biological explanation how microbiome and gut health in general is affected by heat stress. So that can be a biomarker, can be a measure of stress, but also a more resilient ecosystem in the intestine can help the animal to recover from heat stress. So there could be a double use of gut microbiome here, but we're still at the very early phases of research on this topic. At any rate, of course, we have to move forward. So this summer, actually in 3 weeks, we start a new data collection in a sow farm. This data collection will include microbiome and genomics. We actually leverage this on another USD project that we got funded together with Dr. Brito at Purdue University and Dr. Jay Johnson at USDA, still stationed at Purdue, West Lafayette. And so we'll go to a sow farm, again in collaboration with SPG, and we'll phenotype about 1,500 sows for a number of indicators of heat tolerance. In addition to that, we'll do, of course, the genotyping that we need for selection, and we'll also collect gut microbiome so that we'll be able to link these heat tolerance phenotypes to the microbiome composition. And we'll be able to see if the sows that are more heat tolerant have a different microbiome composition. This could lead, for example, to the design of probiotics or prebiotics that allow us to improve the heat tolerance by modulating the microbiome. I'm about to run out of time, so I'll be quick. This study in the commercial farm will be followed by a study on a research farm at Purdue University. Again, Dr. Brito, Dr. Johnson will specifically take care of this. We are going to use a lot of the precision livestock farming tools that have been highlighted today, for example, cameras for measuring, for monitoring behavior and so on. And we'll also dive again into the use of microbiome even for the gut and vaginal microbiome, because in this case we'll be able to heat stress the pigs on purpose, and so we hope that we'll gain further insights and more precision from this study and identifying what are the species that can serve as biomarkers of heat stress. Of course, we have the part, and I'm going back, on the commercial farm, because ultimately we need to deliver something that can be used in the industry. So we need to work on a commercial farm and implement gut microbiome collection in that farm in order to be able to have final proposal for the industry. I am up to close, so I will just summarize on the potential uses of gut microbiome for production. We are very, very at the early stages of research for what it concerns the use of gut microbiome. So we still have room for some creativity and speculation here. But allow me to say that, first of all, gut microbiome, I see that as being as a biomarker. When we have a very expensive trait that we can't really measure, that just think about meat quality, right? Intramuscular fat or meat tenderness. Well, we might just get a sample of the microbiome of the pigs. It's cheap, it's quick, it's inexpensive, and that could turn very useful. If we could show that it works also for heat stress or stress in general, I think that would be very useful, because with a rapid and inexpensive method, we could have on a large scale a measure of stress, a very detailed measure of stress. The gut microbiome also provides some further biological understanding. And let me give you an example outside of the breeding. Let's say there's a research trial and we see that by changing the diet, we see also a change in some commercially relevant traits. Well, gut microbiome is something that could sit in between, because the diet will alter the microbiome, which in turn will alter the commercially relevant trait. While this may not be so important from an implementation standpoint, or maybe yes, but anyway, it gives a further understanding of how microbiome and gut health in general affect the commercially relevant traits. Last but not least, I have to close with this because, again, I'm a geneticist. The gut microbiome can be used as a correlated trait in selection, which kind of brings back the idea of using microbiome as a biomarker. There are some analyses I'm just running right now. I didn't have results ready. But microbiome could replace, in some cases, the genotyping of the individual. Basically, instead of genotyping the individual, or actually instead of measuring some expensive trait in the animal, once again, meat quality or heat stress, we just take a sample of the microbiome, of the fecal microbiome. Again, it's quick, it just takes a swab. It doesn't need trained professionals. And with that information, we can improve our breeding programs in a way that it's cost effective and efficient.